here.
Yeah, live. Yeah.
Okay. I can start, yeah? Right. Okay. Assalamualaikum, everyone. Good afternoon. So we are now in the first invited session, session 1B, which is in parallel with another session 1A. Welcome to ASOMPS, and I'm very happy to be here with very nice guests. Some of them I know quite well. Together with me, I'm Professor Fuzira Shahari, and with me today I have my young co-moderator, Dr. Mumtaz Abu Bakar. We'll be with you for the rest of the afternoon. Okay. So I think today we have 4 speakers, a lineup of 4 excellent speakers from across the country, across the world. We have from Korea, from Indonesia, from India, and also from, I think it's Canada, very far away. Okay, so without further ado, I will start this session with our first honored speaker, Professor Cho Hee Kim. Before that, Professor Cho Hee Kim, let me read some of his biodata. Prof. Kim comes from the biology department, Cheongnam National University of Korea. He obtained his PhD in Osaka University, Japan, and spent his early research days in National Institute of Health in the US. His expertise is in neurodevelopment or developmental genetics. He has published more than 200 papers. Presently, he is the director of Zebrafish Center for Disease Modeling in Cheongnam University. He's also on the medical advisory board member of rare neurological diseases. He is much renowned for his work on development of zebrafish disease models for biomedical and drug developmental research. He has established various neurodisease models such as in autism, spectrum disorder, Lou Gehrig's, Mars Cancer, etc. You will hear in his lecture afterwards. And his title of the talk today is Functional Validation of Mental Disorder Genes Using Zebrafish. So Prof. Chiu will give his lecture for 25 to 30 minutes. And if anyone has any Q&A, please put your question in the chat box via Slido, and then we will attend to those questions later. So without further ado, please go ahead, Prof. Kim. Thank you.
Okay, thank you, Gojira, for invitation to this very nice the symposium.
Perhaps you can share your screen.
Okay, I am here in South Korea. And additionally, I have a friend in the UPM, including Gojira and Mida and Munira, Sajel. I'm using a zebrafish to study the mental disorders. First, let me briefly introduce what is the mental disorders. Additionally, as you can see here, there are so many mental disorders, more than hundreds of the mental disorders. And as in a subgroup, major group like this, the anxiety, the mood, and the schizophrenia, and the dementia. Actually, the Alzheimer's disease is just one of the dementia, and eating problems. First, I want to emphasize the recent progress in DNA sequencing technology. After 2001, the Human Genome Project, for last 20 years, there was a really the big progress. I can say more than 1 million times progress in this technology. I have special interest in the autism, and nowadays we call this as a spectrum, again, because there's so many types of and different type of the autism. This is listen to the genomic data. First, you have to see the numbers in the bottom. If you the sum of these numbers will be the more than 30 million people. Okay, they use the genome data with this number of the people. They made the conclusion the autism is mainly caused by genetics. And again, here one concept of some ignorone, saying the ignore plus the genome. Now that we have the many information, really big information about the genes involved in mental disorders. This is the simple the summary saying that most of people in the world actually focusing on just a few of the known genes, but there are so many of the new genes. Okay, especially for the drug development, you have to the focus or have interest in the new genes. However, there is a really big gap between the information, human genome information, to the final drug development. To bridge this gap, we need the model animals. I'm using the zebra fish. And why we use this? Because this is very rapid and cheap. Okay, you can easily get the large numbers of the embryos, and the development is really rapid. This is just day one. You can see the eye, brain, and if we touch this animal, they respond and they move. Because the fish is a vertebrate like the human, so here is direct the comparison between the human protein and the zebrafish protein. As I can say, almost the same. I start to introduce the concept of the Lego concept. If the individual genes between the zebrafish and the human actually are the same. We have the same Legos. However, the combination of these Lego blocks, we can make a different shape of the animals from the fish, the mouse, and human. This is the basic concept to understand the DNA genes and the genomes. I actually added this new picture. This is now under the revision by a journal. Additionally, we are testing the neuropeptide. So here, okay, there's the melanocortin receptor and this hormone. Here's we just got this modeling. That I want to say that basically the structure, the protein structure, especially in the ligand and the binding pocket is very similar between the human and the zebrafish. And the hormone, see, this is the human hormone and the zebrafish hormone, the 100% same between these two different species. And melanocortin is important in say the hair, the color, and the appetite control, and the obesity, and epilepsy. And just yesterday I got this information. So one of the derivative was already approved by the US FDA as a drug. Okay, here's one easy example using zebrafish to test some bioactive compound. This is the zebrafish, the embryo at day 2. You can see this is the size is just several millimeters. On the microscope, we can see this the pigment cells, the melanocyte. And if we treat here, simply okay, the melatonin or the adrenaline or even a coffee, okay, we can test those the bioactive the chemicals. In this case, if we treat the MCH hormone, it responds in just 2 minutes. And with this assay, because the animal is the very small, we can screening in large numbers. We can directly use the 96 the microplate. In this case, actually, we tried to screening anti-epilepsy drugs by using the chemical-induced epilepsy model. Here's one example. We screened actually this number of chemicals. These are actually a representative of the chemicals. We have the chemical bank in Korea. Thus, we isolated several interesting chemicals. Here's another case. We use the zebrafish at day 1 because they started to move like this. This is the first movement. We call this just a simple tail calling, meaning they have the muscle system and the motor neuron system. And here we are testing a chemical, the ivermectin. Actually, it was a general, actually, the anti-parasite actually in the world. And if we treat with this chemical, they don't move. However, there was heart beating was normal, and this was really active. Just it worked at this concentration 0.1 micromole. Now I have information. Actually, this chemical work as a glycine receptor agonist. And I was surprised to know this chemical was originally isolated from the bacteria. And Dr. Satoshi Omura, he won the Nobel Prize several years ago. And all these, the scientists actually developed the drugs from the natural product, like you. And interestingly, this chemical is also interested as some the possible drug against the coronavirus. So we have to be very careful in using this. This was just reported that this month. Anyway, we can test this chemical in a very easy way in zebrafish. Okay, back to the mental disorders. This is the zebrafish brain. I can say, okay, zebrafish has also the similar brain structures compared to the mouse and the human. And fish, actually, during evolution, the fish start to have this mammalian structure in the brain. We have the transgenic system to monitor the mammalian structure in the brain. The most important character is the fish has this kind of the behavior, the shoaling and the schooling. And this is very important to understand the social interaction, because we use the zebrafish to understand the autism. So we established that this test years ago. So we put their friends in one side, and we put a test fish. As you can see here, they show the very high level of the social interaction. However, in this case, we used the autism model. We made a knockout of the autism gene, DYRK1A. The fish, the shape looks normal. However, in this social interaction test, okay, they don't respond. They don't show the social interaction with his friends. And this is another way to see the social interaction, is the shoaling behavior. This is the model for the schizophrenia. Okay, we also made the knockout mouse. That we can measure the distance between the individual animals to measure their degree of the social interaction. And we have the transgenic system to monitor the individual neurons in the brain in a live animal. Okay, this is a calcium report. This was induced by the chemical-induced the epilepsy condition. Okay, simply we can say this is the neuronal cell fire. Okay, this is the 1 minute and 10 seconds, and the 13 seconds you can see a kind of a rhythm in the brain. This is comparable to the human. Okay, the PET things. However, if we think the resolution this system is 100 or 1,000 times the high resolution. And in collaboration with Professor Youngju Yoon and KAIST, recently he developed a new technology. We provided this animal, and he developed the new microscope. He is calling this the 3DM like that. Okay, this is still in live animals, live brain, that we can measure the activity of individual, the single neurons in the brain. Okay, this the forebrain, this is the optic tecton. Okay, we can monitor the neuronal firing in a live animal. And then here we can treat whatever the chemicals or natural compounds. And we basically, okay, we have very easy animal model, and we have the advanced molecular technology, so-called the CRISPR, the gene knockout technology from very early stage. And very fortunately, we had a collaboration with a human patient, with human genetics people in the Harvard Medical School from 2009. Okay, this I briefly showed those with a simple animal and the new knockout technology and the human patient information. We reported the many cases. Basically, the one every year. Actually, last year we reported 2 cases and 1 case, and this year here, this is all these are actually new type of the patient syndrome involved in mental disorders or the autism or here the ataxia like that. We also have models for the Lou Gehrig and the schizophrenia and the bipolar disorder too. Here's one case. This was reported 2015. Okay, simply look at epilepsy. Just in the previous speaker, Professor Laman introduced about the epilepsy. Here we have the new type of the epilepsy. Okay, see, several generations, how it was difficult to find the causing gene. Okay, it took several generations. Okay, but during the time there was really big progress in DNA sequencing technology. Now, okay, they sequenced all of the family members, and they found the causing gene. This one in X chromosome. They informed us about this gene, and we made a knockout zebrafish. Here, again, day 5, animal, eye, and the pectoral fin. You see the heart beating here. See the eyeball movement. See, the fish actually can start to have the visual activity from day 3. However, in the patient, the animal, okay, they show very hyperactive movement. This pectoral fin is the counterpart of the arms. However, there was no eyeball movement. Okay, I was too surprised with this phenotype. And if we see the jaw movement, they have very rapid jaw movement. This is very similar to the symptoms of the epileptic patient. Okay, the seizures, and they don't move the eyeball, and they have problem in simply on their bubbles in their mouth. We also made the knockout the mouse, shows the similar phenotype. And if also we have the new target, we can try to find another one with this kind of the molecular biology technology. We call this ISA2 hybrid screening. If we have one protein, we can find the finding another one. Thus we found one, and we made a knockout. Here, this wild type at day 5, having the free swimming. However, this patient, they don't move, but there's heart beating, others look normal. And this is a model for the so-called ALS, is the famous Lou Gehrig disease. Okay, this gene is actually a candidate for the Lou Gehrig disease. Here again, thus we made a knockout mouse. This is most the typical assay, just a tail suspension test. The patient animal can have a problem in the muscle movement and their locomotion too. The simple I can explain all these phenotypes, I introduced epilepsy and look at it. See, the phenotype looks completely the opposite. However, now we have the molecular information. These genes are actually involved in the development of a switch in simply in two ways: simply the minus way or the plus way. Okay. That's anyway we have new information. Understand the two. Now we call all these the movement disorders. Here see the Parkinson's disease, the ataxia, the chorea, and the Huntington's disease. Okay, now we can understand the movement disorders at the molecular level. From on the basis of these results, okay, because we know about the gene, then if there's a similar patient, we can easily confirm by the genomic DNA sequencing or just by simple PCR, and we established the patient foundation several years ago, and the last year the similar patient the foundation was established. And I want to introduce another case. This is my favorite gene in the scientific life. So we reported that this is a new chemokine or cytokine neuropeptide novel gene. Okay, this gene is expressed in the very specific region in the brain. So we call this the habenula. This brain small region is involved in emotional control and involved in the emotional pain and the stress, anxiety, and many things. This is very important to the target, and we made a knockout. So if we put the animal in new environment, they if they have the very anxious in the new environment, the fish stay in the bottom. However, after 10 minutes, they feel easy and they can move around. However, the knockout of this single gene cause this kind of the phenotype. This is meaning that we have a gene involved in the anxiety. And we also made the mouse. This is a mouse, a test, the anxiety test. They just put the mouse in a high place. They operate something, and the mouse also show the similar phenotype. Because we have a long collaboration with the human genetics people in the Harvard Medical School, and I asked them the finder whether some patients have a mutation in this gene, and then they found the family, and they informed me they are the autisms. That's why actually I started the study of autism about now 10 years ago. And we did actually many approaches at the molecular level, cellular level, or some electrophysiology, many things to understand the molecular action of this new chemokine. Okay, and my final goal is in drug development, and now we are trying to characterize its putative the receptor. And this is so far conclusion is the SAM2 is involved in the control of the CRH, the hormone. This is the key hormone involved in the stress control. Okay, from external signals. We have to decide how to react. Simply we say the flight or the fighting, like that. And finally the cortisol works as the final hormone, and at the cellular level, SAM2 works as the ligand. We have the putative the receptor, because this is the GPCR and involved in the G protein signaling. And next I will introduce this one. This is one of the G protein gamma, the number 8. Okay, PV knockout. Okay, see, very actually quite interesting the phenotype. Okay, this is normal the mouse. separate the big or the red. Okay. However, if we knock out this single gene, okay, they don't operate. Thus, I call this the mighty mouse or super mouse. However, in human, there's a model of the ADHD. We reported this last year in the molecular psychiatry. And okay, because I tried to actually, because this symposium is the most of the people involved with the medicinal plant. This I found a very important the paper is the quite years ago, but they used the zebrafish to test the medicinal plant, and they are saying the mescaline show a very similar phenotype to our knockout zebrafish. Okay, thus I contacted to them, and now the horse is now as a medical doctor in New York. Thus he also had the interest in because they don't know the mechanism. Thus we want to collaborate. Okay. Thus anyway, this chemical come from the small cactus. And I realized there's several other interesting chemicals from mushrooms. And I move on to another case here, ataxia, and in hospital they are saying leukodystrophy. Okay, what is the leuko means the white, and the myelin. Okay, and there's the similar disease. They are saying multiple sclerosis, the Lou Gehrig, ataxia. But to me, at the molecular level, all these actually the diseases have a similar molecular mechanism. I will introduce about this. Okay, we started this with a Korean patient with this age. She had very severe problem in the brain and in locomotion. This shows her MRI, and from the whole exome sequencing, they found the candidate gene, and then they informed us we made a knockout zebrafish, and this is the characterization of the knockout animal. This is day 2, showing the motor neurons. The white region shows the spinal cord, and the branches shows the growing the motor neurons. Up to day 2, they look normal. However, at day 5, this is a transgenic animal showing that the myelin structure. As you can see here, the myelin development is severely affected, and this is very similar to what found in the human patient. And this was a new finding. We also can monitor the development of the vessels in the live animals. There was some abnormal angiogenesis in with these neural disorders. And then we also introduced the proteomics, and we found the new things. And at the molecular level, actually, this gene is involved in the protein translation and unfolded the protein response or the stress at the molecular level. That's okay. That's from the molecular study, we can understand the myelination is a very complicated the process. With protein new protein translation, and what you found from the proteomics. Actually, some of many of patients also have a problem in the eye range. Okay, that's now we can understand. Okay, there's similar the mechanism in the protein process. And this is a summary. Okay, because with again a big progress in genomic sequencing, and here's the many the mental problems, and they found the causing genes. But as you can see here, all these causing genes looks similar. Okay, meaning all these actually genes are involved in the translation. All these are the amino acid tRNA synthase. Okay, and I can say all these actually patients have the similar molecular mechanism, and this is what we found. Okay, the angiogenesis looks involved in on this disease. Okay, finally, I'd like to introduce the successful case in drug development. This is a new patient with the the autism and she has a problem in eyeball movement like this, and then have problem in eye contact. Eye contact is again important in the social interaction and the autism. Okay, and I have a much interest in this one at the cellular level. We call this the central pattern generators. Okay, most easy case is the heart beating. We have a nervous system to control this heart the pace. Okay, and nowadays the CPG actually we have the several CPGs in the brain, okay, in the heart, on the lung and intestine, or in the spinal cord. Okay, for locomotion, we need the CPG. Okay, we made a knockout with this gene. Okay, this is the patient. This day too, if we touch, they just escape or run away. However, the patient can escape, although they show them a touch response. Okay, with this model, we screen some chemicals. Here, see, it's amazing. I was so excited with this chemical. Thus, we are testing. We have a collaboration with the chemistry field. Thus, we are testing and try to develop a really good chemical. Okay, thus we see the geography is useful in the medicinal and the plant and the spices. And if you have good chemicals, please inform me, and we can help to test in this easy animal models. Okay, thank you very much for your attention. Also, I would like to say thank you to my lab members. Thank you.
Thank you, Prof. Kim. Do you mind to unshare your screen so that you can see everyone? I think there is some lagging between our room and participants' room. They have not quite finished. So now we have to wait a few seconds, I hope. By the way, that was a very impressive lecture. I'm always impressed with the work that you've done with your team. And I think there are a question I saw in the chat box, so perhaps I would read it out now. Okay, I think yeah, there's a question by Dr. Intan Safina. Hi, Prof. Kim. Are metals such as lead, plumbum, lead or plumbum could give a very significant inflammatory effect on the zebrafish brain? To answer that, or maybe I don't know. But I think you can go ahead and answer.
Sorry, what was the question?
Yeah, the question is, are metals such as lead, can these metals give very significant inflammatory effects on the zebrafish brain? Lead, plumbum, PD.
Prof. Kozi, I think that question is for the second presentation. I think maybe, is there any drugs?
It's meant for Prof. Kim.
Oh, okay.
So this is to do with lead poisoning?
No, still I couldn't get the point.
Yeah. Well, does it cause any inflammation?
Inflammation?
Yeah, inflammation on the brain.
Yeah.
These, yeah.
Yes, I have much interest in inflammation because nowadays for the schizophrenia, that actually many of the brain disease is involved in the inflammation. Thus, I have much interest in inflammation. Like now, there is the controversy whether the inflammation started within the brain or come from the outside. But nowadays, people are the more interested in the actual inflammation from outside, from the whole body actually. This is one of my the major concern. Thus, that will be very interesting Okay, anyway, the implementation is very important, but we don't know much about that. Yeah.
Right. So there's also one more question. I think I can only entertain 2 questions. I think the remaining other questions you can post to Prof. Kim to the chat box, to his chat room. The last question for you today is there any drugs or medicine to cure autism, especially from natural product molecules that has an effect on autism from your work? Okay, Prof. Kim? Get the question.
Sorry. Okay. What was your question was my chemical?
Yeah.
Was
Is there any that is effective against autism at this
yes,
level? At this
yes, sir. For that, for the last one, the patient is actually the autism, the patient. In that case, that was the chemical. Actually, that was now I cannot tell about the chemical, but that was quite a general chemical, very easy chemical. Yeah.
Okay. Right. I think since time is limited, we need to move on to the next speaker. That has been a very enjoyable lecture. I am very impressed, and I think you and your team has made a huge contribution to this area of orphan disease. And we'd like to congratulate you and wish you the best in the future. Thank you.
Thank you very much.
Now I hand over to Mumtaz for the next speaker. To introduce the next speaker.
Okay, thank you, Prof. Kozira. Okay, so our next speaker is Dr. Ati Vidya Waranti. She's from Faculty of Pharmacy, Universitas of Erlangga. She is currently head of master program and also head of Nature Product Medicine Research and Development in Erlangga Universities. Okay, her major research are research and development of herbal medicine product, finding leads compound from natural sources for an anti-malaria drugs, phytochemical analysis, and quality control of herbal medicines. She published in 69 international journal with corpus citation of 770 and H index of 16. Today, Dr. Ati will give a talk on prospect of Andrographis paniculata, herbal product for the treatment of malaria in pregnancy. So without further delay, I would like to invite Dr. Ati. So Dr. Ati, the mic is yours.
Thank you very much to Dr. Mumtaz Abu Bakar for the time, and also thank you for the Professor Kozira Binti Sari. Nice to meet you. May I share my
yes, doctor, you can
presentation?
Share.
Yeah. Okay. So wait, how to slideshow this?
You have to press the slideshow on your computer.
Yeah. Sorry. Okay. Okay. Okay.
Yeah.
Okay. Thank you very much. Good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to say thank you to the MNSP President Professor Dr. Nur Hadiani and the Chairperson Associate Professor Dr. Intan Safina and the committee for inviting me to this fascinating conference. This afternoon, I will deliver my presentation, which talks about the prospect of herbal product from Andrographis paniculata for the treatment of malaria in pregnancy. Malaria is still a major health problem in the world. WHO reported that in 2019, around 229 million cases were estimated, and with 409,000 deaths for malaria. And in Indonesia, there are 23 cities that are highly endemic for malaria, and around 225,790 malaria cases. Children aged under 5 years and pregnant women are the most vulnerable group affected by malaria. Around 11 million pregnant women exposed to malaria infection in 2019 delivered about 972,000 children with low birth weight. And pregnant women are a high-risk group infected malaria, particularly susceptible to malaria infection due to immunological changes occurring during pregnancy. And the causal parasite of malaria, Plasmodium falciparum, avoids detection by the maternal immune system by occupying blood spaces of the placenta. Pregnancy-associated malaria or placental malaria is characterized by sequestration of Plasmodium falciparum and infiltration the immune cells within the intervillous space of the placenta, and the sequestration of parasite-infected erythrocyte in placenta will activate the inflammatory cytokines, resulting in impaired maternal fetal exchange and damage to the placenta. The effect of malaria pregnancy on fetus could be abortion, stillbirth, preterm delivery, low birth weight, neonatal mortality, congenital malaria, and also infant mortality and anemia. Several medication has been established for treatment malaria in pregnancy, including chloroquine, quinine, sulfadoxine, pyrimethamine, mefloquine, chlorproguanil, Dapsone, and amodiaquine. However, increasing resistance to these agents has led to a need for more effective malaria treatments, especially for women pregnancy. This is the example for the malaria drug that has been resistant. First is chloroquine. Chloroquine discovered in 1930s and widely used from 1950s until 1990. And resistance started independently in Colombia and Thailand. Sulfadoxine and pyrimethamine, it is a two-drug combination introduced after CQ resistance had taken hold, and the resistance evolved rapidly and now occurs in high frequency in major malaria regions. The first line anti-malarial drug, artemisinin in combination, recommended by WHO for treatment malaria since 2001, and now resistance established in parts of Cambodia, Myanmar, Thailand, and Vietnam. The difficulty of malaria therapy in pregnancy is more often caused by the selection of safe anti-malaria drugs for mothers and fetuses, because most of the available anti-malaria drugs provide side effects for mother and fetus. Some anti-malaria drugs are contraindicated also in pregnancy, and some may cause severe adverse effects. For example, mefloquine has been associated with an increased risk of stillbirth. Sulfadoxine and pyrimethamine is safe but may be ineffective in many areas because of increasing resistance. And primaquine and tetracyclines should not be used in pregnancy. Amodiaquine, chlorproguanil-dapsone, halofantrin, lumefantrine, and piperacin have also not been evaluated sufficiently to permit positive recommendation. WHO recommends artemisinin-based combination therapy especially in the second or third trimester of pregnancy for women with uncomplicated Plasmodium falciparum malaria. Unfortunately, information of the safety, efficacy, and pharmacokinetics of artemisinin-based combination therapy in pregnant women is limited, and Directorate of National Vector-Borne Disease Control Program in India does not recommend artemisinin compounds in any trimester of pregnancy. Therefore, there is urgently need to discover new safe anti-malaria medicine for pregnant women and babies. The use of plant in anti-malaria drug discovery effort has been widely practiced, particularly from herbs traditional use by communities to treat malaria. The ethnopharmacological approach can be a promising venue for finding the effectiveness and safety of novel anti-malaria candidates. The most important anti-malaria compound has been isolated from plants. They are quinine and artemisinin. Quinine obtained from cinchona bark, and it remains an important anti-malaria drug almost 400 years after its effectiveness was first documented. And two leading derivatives of quinine is chloroquine and mefloquine. How about the artemisinin? The artemisinin was isolated by Chinese scientists in 1972 from Artemisia annua. These compounds contain androperoxide bridges, which is essential for its anti-malaria activity. And the derivatives of artemisinin that has been used until now is dihydroartemisinin, artemether, artiter, and also artesunate. In Indonesia, the plant, which is known as sambiloto, and in Malaysia, people mention hampadu bumi, is used as a traditional medicine to cure malaria disease. Andrographis paniculata belongs to family Acanthaceae, one of the most popular medicinal plants used traditionally to treat several diseases such as cancer, diabetes, high blood pressure, ulcer, leprosy, bronchitis, and also malaria for centuries in Asia, America, and Asia, African continents. Our previous study from 1995 until 2011 has been found that Andrographis paniculata is active as anti-malaria. And anti-malaria and immunology activity from Andrographis paniculata has been reported, and many articles has been informed about this activity. For example, the methanol extract of Andrographis paniculata have anti-malaria activity and non-cytotoxic at high dose at 100 milligrams per kg. And also the major compound of Andrographis paniculata, andrographolate, was found to have potent anti-plasmodium activity against Plasmodium falciparum and Plasmodium BG strain angka. And for the activity immunology, Andrographis paniculata extract and andrographolate show several anti-inflammatory activities: suppression of inducible nitric oxide synthase, reduction the interferon gamma and interleukin-2 production, inhibition of TNF-alpha, and activating of NF-kappa B. Andrographis pandiculata, as we know, contains several active constituents, include flavonoids, flavonoid glycoside, and lactones and diterpenes. And the major compound andrographolite was isolated from the aerial parts, including other lactones. Neo-andrographolite, deoxyandrographolite, and 14-deoxy-11, 12-dihydroandrographolite. We have been developed a standardized herbal product from Andrographis paniculata. This tablet contain ethyl fraction of Andrographis paniculata equivalent to 35 milligram of andrographolite per tablet, namely IS20101, and several tests to evaluate the anti-malaria activity of this product has been done. It was found that this product has anti-malarial in vivo against Plasmodium bergei infected mice with effective dose 50. 6.91 milligrams per kg, and it's classified as a highly active anti-malarial substance and able to increase survival time of the mice infected parasite. We have been conducted a study to determine whether this EPI product has the prospect of being an anti-malarial pregnancy associated malaria. We evaluate the EPI product activity in inhibiting parasite and reducing the activity of cytokines due to parasite infection in the placenta, which can protect of the fetus from damage. This is the following slide is a simple schematic illustration of the immunology of placenta malaria, especially the cytokines that we have evaluated. The attachment of Plasmodium infected erythrocyte to the chondroitin sulfate A, CSA, CSA receptor in the placenta may causes parasite secretion in the placenta and resulting in placenta malaria. This condition will increase the level of pro-inflammatory cytokines from Th1, such as interferon gamma, TNF-alpha, and also will increase the anti-inflammatory T helper 2, such as the interleukin 10, which are the interleukin 10 down regulators in macrophage. And also the increased interferon gamma cytokines will affect the secretion of TLR4 and NF-kappa B, which in turn causes the apoptosis. And also the TNF-alpha will increase, the increase of TNF-alpha will also affect the secretion of COX-2 and prostaglandin A2. As we know, it is the pro-inflammatory cytokines that it causes the damage to the fetus. For example, the low birth weight, spontaneous abortion, premature delivery, and also abnormality and morphology. We start the experiment with mated the female and the male mice in monogamous pairs until we have the successful pregnancy. The pregnant mice were divided into 4 groups, and all mice except the normal groups were infected by Plasmodium bergei parasite on day 9 of the gestation, and were given therapy on day 11 gestation. And on day 15 of the pregnancy, all mice were sacrificed. The placenta expression and to observe the morphological abnormalities. So this slide is demonstrated the administration of IP products could reduce the expression of chondroitin GSA, as we showed from this figure. This is the C is IP product, and CSA is a unique receptor that binds to parasitic infected erythrocyte through the Plasmodium falciparum PFMP1 in the placenta. Decreased CSA expression will cause the binding of PFMP1 to fall and resulting in reduced addition of infected erythrocyte in the placenta, so that ultimately lowers parasitic abundance. So in this experiment, the CSA expression of mice infected Plasmodium bergei treated with epi product lower than DHP tablet. DHP tablet is meaning dihydroartemisinin and piperacin. Another experiment which show that the accumulation of the parasite on placental tissue can lead to pro-inflammatory immune responses and tissue damage. Then the administration of this product twice daily for 4 days decrease parasite in placenta. Also, this product can activate the TGF-beta expression compared to the untreated. The transforming group factor beta or TGF-beta is a cytokine regulator of inflammation that important in inhibiting parasite growth and preventing inflammation. The administration, the activation of TGF-beta expression in the placenta could clear the parasite and prevent inflammation and pathology that cause adverse effect of the mother and fetus. The placental malaria may cause the increase of the TLR4 expression, and the product could decrease the TLR expression and apoptosis index of placenta, and thus increasing fetus complications such as abortion. So in the placenta, apoptosis occurs typically to maintain a pregnancy, and excessive increase in apoptosis may cause a disruption of blood flow to the placenta, resulting in decreased fetal weight. This is the table that the epi product can reduce the TLR4 and also apoptosis index compared to the untreated group. In this slide, we also show that the epi product decrease the COX-2 and placenta prostaglandin compared to the untreated group, and the decline of COX-2 and prostaglandin could prevent premature abortion and labor and pregnancy malaria. This experiment also show that the epi product can decrease the TNF alpha and interferon gamma expression in the Plasmodium bergei infected pregnant mice. the expression of TNF-alpha is lower than the untreated group. Also in the interferon gamma expression, the epi group is lower than untreated group. It is also epi product can decrease the interleukin-10 expression. So it is the decrease of the TNF-alpha and interferon gamma, and also regulate by the decrease the interleukin-10 expression, can protect the fetus from the damage. This slide shows you how epi product can protect the fetus from being damaged by the parasite during pregnancy. The untreated group and the HP group show morphological bone abnormalities, as we shown in the figure 1. In the cranium, there was only os nasal, premaxilla, frontal, and parietal had a size larger than normal. In vertebra, it was seen that the formation of vertebra is not perfect, as shown in the figure 2. And in the costa, which look shorter, malformation and broken. Skeletal from legs and hind legs, namely on scapula, humerus, radius ulna, femur, and tibia, fibula, also seem to be shorter than normal. And from the active group, we can found the information that epi product didn't cause the abnormalities in fetal bone morphology. Cranium, scapula, costae, and vertebrae, humerus, radius ulna, tibia, fibula looks normal and similar with the normal group. So this study also showed that the administration of the first-line anti-malarial drug, dihydroartemisinin, piperacin, in the first trimester of pregnancy could cause fetal damage in pregnant mice infected with the parasite, so that the use of DHP in pregnant women needs to be considered. So from this experiment, from this study, we can conclude that the herbal product of Andrographis paniculata can inhibit the growth of parasite in the placenta, thereby decreasing the expression of pro-inflammatory cytokines that play a role in malaria infection in pregnant mice. Furthermore, it can protect the fetus from damage caused by malaria parasite infection. We expected that this herbal product prevents the fetus from malaria infection during pregnancy, probably due to its primary compound, andrographilide. And therefore, this herbal product of Andrographis paniculata could potentially used as an alternative and complementary medicine for curing malaria in pregnancy. I think it is all of my presentation today. Thank you very much for your attention, and also thank you for the team in University of Erlangen. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh, Dr. Ati. Thank you very much. It's a very interesting topic, it's a very interesting talk. So I would like to see if any questions for Dr. Ati. Let me check on the chat box. Okay, there's no question because they have a delay like 2 minutes, around 2 minutes.
Oh, I'm so sorry.
That's why That's no. I mean, like, it's not on your part because the one that participants view is like delay for 2 minutes, I think. So that's why. Okay, now it's end. So maybe they have a question. So it's okay. Dr. Ati, is it this research is already be like developed, fully developed, or it's just like just starting or just the preliminary ones?
Yeah, thank you very much, Dr. Muntas. I think it is very preliminary, especially in malaria in pregnancy. But from this experiment, from this study, we can continue to research the how is the product of Andrographis paniculata can use to cure malaria in pregnancy. Yeah, so we have to continue to the clinical study, especially in pregnant women. Yeah.
Okay, so this is, I mean, like, it's very interesting because we know like malaria is very dangerous in some
Yes.
yeah, so especially for the pregnancy woman, right? So it's very interesting. Anyway, so let me see. Okay, there is no any questions from the audience. Okay, so
There's one
sorry,
question in the chat box.
Professor Kozira.
of course, I can't see the question on the chat box.
Okay.
Can you please read for me?
I'll read it. Okay. This is a question from student Siti Mariam from the ITM. May I know how long is your research on this Andrographis paniculata from plant to the tablet and to the product?
Yeah. It is very long time. Maybe from the 1995 until now still do the research, but maybe it is due to our funding in Indonesia and in our university is not much, so we have to waiting for the grant. But actually, if we want to study about the anti-malarial drug from the laboratory until the clinical, yeah, it is not easy, I think, because we have to study about the toxicity besides the efficacy. Yeah, toxicity in animal, also toxicity maybe in a human. So it is a long journey, I think. So it is not finished. Still continue to doing the research for the goal of the Andrographis paniculata can use in human for the cure the malaria, especially in malaria pregnancy. This maybe is still started. Thank you.
Okay, thank you, Dr. Ati. There is another one question here. Thank you, Prof. Kozira. Now I can see the chat box. Okay, Dr. Atif, thank you for the informative presentation. What is the concentration of each tablet and how many tablets to take? And is it for prevention of cure or for the cure?
Oh yeah, but Andrographis product is per tablet is contain at 30 milligrams andrographolite for the standardized. So in this study, we used around 100 milligrams, use the ethyl acetate fraction, or use the tablet, and we counting the andrographolite doses as 100 milligrams per kg of body weight mice. Yeah. And what's the next question?
The next question is okay. How many tablets to take? And is it this table is for prevent or for the cure?
This is for cure. So I have I have done the clinical trial in people in Maluku Island, for the not for the pregnant woman, but for the non-pregnant, and also for the another people from age 18 until 60. So it is we use the for 2 times a day and per day are 2 times 4 tablets 2 times a day. Yeah, for 5 days cure.
Okay.
Okay. Yeah.
Okay. 4
Then
tablets.
yeah, 4 tablets 2 times a day for
Okay.
consecutive 5 days.
Okay.
Yeah. So we have found that in their endemic area, this tablet could inhibit the parasite. We evaluate the parasite from the staining Gamsa staining thick blood. Yeah.
Okay, Dr. Ati, thank you very much. Okay, since we are running time, so I think thank you very much, Dr. Ati, for very interesting, very informative talks. Okay, so maybe I will give to Prof. Kozira to introduce our next speakers.
Okay.
Prof. Kozi.
Thank you, Dr. Mutas and Prof. Kozira. See you.
Yeah.
Thank you. Okay, can you unshare the screen?
Okay, I will unshare. Yeah.
Thank you. All right, our next speaker comes from India, Dr. Syed Ahmad. Okay, let me go a little bit of his biodata. I think he has we can access the biodata in the web page, and then to know more about Professor Syed Ahmad. Well, Professor Ahmad is actually working at the Department of Pharmacology and Phytochemistry, School of Pharmaceutical Education and Research in Jamia Hamdard, Delhi. He holds a PhD in pharmacology and phytochemistry. He spent some years of his young postdoctorate life at Albert Einstein College of Medicine in New York, and he visited several universities around the world to get his experience. He has accumulated more than 300 publications with credit and has also authored several books in the area of pharmacology. And his areas of research interest centers around natural product chemistry and multi-platform metabolomics for applications in targeted and untargeted metabolite analysis in biological systems and the quality control of novel drugs. His talk for today is metabolomics in quality control and drug development from botanical to traditional medicine. So over to you, Dr. Syed, Prof. Syed.
So, good afternoon, Assalamu alaikum. So, can Dr. Kozira, Professor Kozira, Dr. Mumtaz, can you hear me?
Yes,
Yeah,
we can
hear you, but very, very low. Can you increase the volume?
Yeah, so is it okay right now? Yeah, I increased
A bit
volume.
better.
Yeah.
A bit
So,
better. Yeah.
Can you see my screen? Is it visible?
Yes,
Yep.
we can see.
Yeah. So should I start now?
Yes, please.
Yeah. So thank you very much, Professor Kozira and Dr. Mumtaz and organizing team of this program, and Professor Ibrahim Jantan, my friend from Malaysia, for invitation for this program. So title of my talk is like metabolomics in quality control and drug development from botanicals and traditional medicines, and as introduced by Professor Kozira about my this brief introduction about my work. So we are here in Delhi, situated here in the heart of Delhi, and my institute is Jamia Hamdard, and we are among top 20 universities of India, and this is this university was developed by one Unani physician, and he was the owner of Hamdard Laboratories, in fact. Okay. Then this is the School of Pharmacy from where I belong, and this is my laboratory where I have these facilities and we are working. So we start with the metabolomics, and everyone knows that what is metabolomics right now, since it came for the first time in 1990, and the definition that this is the analysis and our identification of all metaboloids present in a living organism or any system. So then why we are doing metabolomics, since it is going to correlate with the genotype and phenotype and many of methods involved in that metabolomic profiling, like sample preparation and method identification, data collection, and then determination, various statistical analysis. Different analytical techniques we are using in metabolomic profiling, like NMR, HPTLC, FTIR, GCMS, LCMS, and UPLC-MS. Mainly we are using here in my research, mainly HPTLC, GCMS, and LCMS-MS. Okay. So here are some limitations that you can see with the different this techniques, and mostly the NMR is the most frequently used technique, but still we are using others. Okay. Then there are, as I told, that in workflow that we are going to prepare the sample and analyze it and do data analysis with the statistical softwares and like PCA and others, and that is, so there are different type of metabolomic profiling that we are doing. So in metabolomic profiling that can be used for metaboloid profiling, the metabolomics, that means for in fact dereplication and identification of metaboloids present in them, in any organism at a particular time. We can analyze all those, like here I have shown that the data and then its analysis. Okay, so like different data set, big data type, we can collect and we can analyze with different multivariate analysis and come out with some result. Okay. And we do the PCA and analysis, in which we found various metaboloids in different cluster and in quadrant, and that is going to show the differentiation in present in different metaboloids. We can see, we can use metabolomics in authentication of plant material by like if we have same plant like Imblica officinalis from different geographical region, then you can see the differentiation here in their on the basis of their metaboloids, which are statistically correlated, and then we can find this type of that which one is like ZP is closely related to ZD metaboloids. So similarly like that we can find out the differentiation, variation in different cultivars, as I told in before this. So in different cultivars also we can find out the variation utilizing metabolomic profiling. And similarly comparative metabolite profiling of varied parts of the plant, like if it is a oleogum resin obtained from the this stem of Google, then we can take the extract of bark of a stem, then a stem, and then oleogum resin, and find out the differences in them. And then metabolomics in identification of differential metaboloids, like in diseased state and in normal state. we have some metabolites which are increased and some are decreased. So we can find out the differential metabolites using metabolomic profiling and using differential metabolites. Those can be used for prediction of disease and disease status, and even in testing of presence of disease in many cases. And we can use this in pathway analysis also, and similarly using network pharmacology. And then we can predict the mechanism of action if it is going to be used for human purpose. Then toxicity, we can reveal its toxicity by doing in silico that ADME analysis, okay, and we can find out finally the therapeutic effect of a medicine. So that's how this is related to us, and we can find out the pathophysiology of any disease using metabolomic profiling. Okay, so many aspects are there. And even I told that with with network pharmacology, we can find out actual mechanism of action of these metabolites which are present in the plant. So we are going to take a few examples to see that how this is going to related to us, and how this is going to related to medicinal plant, how we can utilize it for the purpose of drug discovery. So see that one example of a flower, Butia monosperma, was used in India, is still used in some region, and by diabetic patients. So what they were doing, and it was used by traditional healers, in fact. So they were taking this flower and then crushing it and boiling it in hot water and taking the decoction. So we thought to work on that. It was working well there, and it was very popular. So we prepared the extracts as per the traditional healer were preparing. It did the toxicity evaluation, found that this was this non-toxic up to 10 gram, and then prepared this, did and in some in vitro models that anti-diabetic activity, and in cell lines as well, found that this was active. Then we prepared the fractions, that is on the basis of polarity as well as on the basis of metabolite. We fractionated like in hexane and in DCM and in butanol and then remaining in water. So like and similarly we fractionated and differentiated the glycoside portion, alkaloid portion, terpenoid portion. So all those portions. So then again we tested to find out which portion is most active. Then we did the metabolomic profiling of each fraction and the most active fraction as well, and find out the number of metabolites by LC-MS and find out total number of metabolites. And then what we did that after introduction to rats, we find out the metabolites present in the rat plasma, did the pattern recognition analysis and find out the bioavailable metabolites. And then we did the yeast cell metabolomics, and similarly gut microbiota and hepatocyte metabolomics to find out if any metabolites were changed by microbiota and present in the blood, or if something is biotransformed by hepatocytes and present in the blood, which is imparting in activity. Did in silico analysis of all bioavailable metabolites, and followed by pharmacokinetics and predicted the activity. So like that we did. So one by one we will list. So we first did the quality control analysis as per pharmacopoeial guidelines using HPTLC, and then followed by HPLC. And then I said that identified all metabolites present in different these fractions, and then in the rat plasma after introduction at different time intervals. And then what we found out of more than 150 metaboloids, we analyzed, we found that these 8 and 9 metaboloids were major metaboloids, showing more than 1% of area. And then we did some other like yeast cell metabolomics to see whether it is showing similar type of metaboloids. So that's also showing those similar metaboloids present in major metaboloids present in the biological system. Then we find out that how much metaboloids in glycoside, alkaloid after 1 hour, 2 hours, like that. We did analysis, differentiated them, and here you can see the summary that in the extract that we used, and when it was containing 27 metaboloids, 15 out of 27 were bioavailable as such, 12 were not bioavailable. Okay, whereas some metaboloids were present as a ghost molecule, which were not present in the original, but these are present. So we analyzed them, find out and identified them. So those metaboloids in fact were obtained from liver metabolism and from microbiota biotransformation. Okay, later on what we did in silico analysis of all those metaboloids, which shows that these 4 metaboloids were having better activity, that is butyrene, isobutyrene, monospermicide, choriospin, luteol, arone, and onion. And then like this, they have better activity in silico analysis. Then we again did HPTLC bioautographic analysis of in the extract as well as on the original compounds of those that reference standards, and confirm that these are having alpha amylase and alpha glucosidase inhibitory activity. We did the pharmacokinetic analysis of them and predicted their activity and proposed the mechanism and published it in some journal. Okay, so like that there are various traditional formulations. One is used like Archimako and Archikasni, that is mainly used here for non-alcoholic fatty liver diseases. So that is in fact the aromatic water prepared from some medicinal plants, and then known as like here Archimako is from Solanum nigrum, and Archicosni is from Chicorium intybus. So they are distillates, in fact these are. So these are used for alcoholic NFLD. So we did that GCMS metabolomic profiling of them. Find out it is containing 56 metaboloids, and when we introduced in the rats, then it is showing 38 metaboloids out of them. And then we did similar in silico analysis and other things. And similarly, like here we are going to discuss many cases. So here I am showing you one Carica papaya, mainly used for dengue fever related thrombocytopenia, and its leaf is used in fact since a long time in India and Pakistan and Bangladesh and surrounding places. So what we did the activity of extract and then fractionated, and then bioactivity guide did fractionation shows that butanol fraction is having the best activity. And then we did analysis of butanol fraction, and analyzed all those metaboloids have been quantified by using HPTLC and LCMS, that is 4-5 metaboloids, which are essential to develop it as a drug here in India. and reduced its dose. Earlier it was 2.5 gram per day in the form of fresh juice and extract, and then it comes to about 500 milligram on the basis of its extractive value, quantified their constituent, that is transferolic acid, caffeic acid, myristotin, and camphorol present in them. Some formulations in India are used, traditional formulations used since long back, and for cardiac disorders, and those are containing coriander and amomum. So we analyzed those formulations for their targeted metaboloids like alpinatin and amomum and coriander and coriandrol and this alpha-panin and eugenol and coriander by using GCMS and HPTLC. Similarly, it also some formulations are also using borago and jetamancy and saffron. So for them also, we did some targeted metabolomic profiling and analyzed by HPTLC, HPLC, and GCMS various metaboloids for their quality control. Similarly, we developed some terpenoids enriched fraction from Bacopa moneri and Picrorhiza kirova as an adjuvant to chemotherapy, since those are, and analyze their terpenoids present in them like cucurbitacins, alpinatin, picrosides, so betulinic acid, so epocyanin. So here you can see that in Bacopa moneri that was showing similar effect. Like here is Bacopa moneri and this is 5-fluorouracil, and it is showing similar effect to that of 5-fluorouracil and the improvement for cancer. Similarly, this Boswellia serrata we used as adjuvant in hepatocellular carcinoma and analyze its constituent. And when we used it with doxorubicin, so it is showing better potential. Here it is this Boswellia serrata extract plus doxorubicin in low dose to reduce its toxicity, that is showing in Culpermer salvivar carb the better potential as compared to the doxorubicin. Then various compounds we screen for diabetes, in which citrulose colosynthese, that one plant is used mainly. So we analyzed its extract and identified the alpha amylase and beta glucosidase inhibitory compounds that is present in this, and identified them. And then similarly, then this Tylophora indica, we prepared some fraction in, and that alkaloidal fraction, and that was active against cancer. And we identified their metaboloids, all those metaboloids present in them. Some formulation like distillate, as I told you, which are most commonly used in Indian system of medicine, and they are having a bigger challenge of, like, when we are keeping them, they are having some precipitation after 2 to 3 months. Then shelf life was the problem. We analyzed all the metaboloids and predicted their shelf life, that when we opened and identified their metaboloids as well, and then predicted after opening the cap, it should be utilized within 15 days. Similarly, like some Morus alba and Morus nigra leaves are used in diabetes as well as in some other respiratory tract disorders. So we identified its constituent, and through HPTLC bioautography and LCMS analysis, and then identified its constituent and prepared in the form of infusion. So we quantified those materials, that DNG and chlorogenic acid and rutin, responsible for its activities. Similarly, picrorhiza and curova and eagle marmalose, so picrorhiza, curova and eagle marmalose has been used in various disorders, but we discussed it since it is mentioned in for diabetes, so for its fraction which is active against diabetes, we analyze its constituent, and then in eagle marmalose, in fact, it is eaglin is, since eaglin is toxic alkaloid present in them, so we use that alkaloid free portion of that, and then tested for that anti-diabetic activity, and the active components were like myricetin, caffeic acid, and gallic acid present in them, and then some estrogenic plants which are present in, we are reported in Indian system of medicine. We did their estrogenic activity in yeast model and identified their components present in that by TLCMS bioautography. Further, we developed some synergy-based combination for learning and memory, and this is in fact just filed the patent and identified their active metaboloids by bioautographic analysis. So in this, what we did, all those plants and medicinal plants are reported in Indian system of medicine for learning and memory. There are about 25 plants. So took all those and tested in vitro and ex vivo models and found that two plants, the most active plants, and then mix them in with isobolographic model, tested again to find out the synergistic combination. In the synergistic combination, we identified the metaboloids which are present and showing the synergistic action. Okay. Then some similar to that, some immunomodulatory plants in this COVID pandemic. We collected all those plants which has been mentioned in Ayurveda and Unani system for immunomodulation, and then tested in different models in vitro and ex vivo system, and like Phyllanthus, black pepper, Tinospora, and Curcuma, Ocimum, Caellia, Vidania, and as a directa indica. Tested them and found that two most active, Phyllanthus and Tinospora among them, and then developed some combination, and even their metaboloids were analyzed and found that how many metaboloids are present in their extracts, analyzed their markers which are common in them, and so like gallic acid, quercetin, ferulic acid, phylamblin, berberine, piperine, bedaberine A, and curcumin. Then developed this synergistic combination out of this also. Similarly, we did some work on management for CKD, since there are many patients on this CKD here in India. So we developed some formulation for them utilizing the medicinal plants which have been reported for kidney disorders. We tested them in vitro conditions and in ex vivo condition in the cell lines, and found these Tinospora cardifolia and Borrelia diffusa were most active, identified their antioxidant compounds, and then we did some network pharmacological analysis to identify if these are going to have some effect on the genes of kidney disorders. Then I identified the targets and found their effect and in silico conditions. And similarly, we did some work on metabolomic profiling and molecular mechanism for identification of non-polar metaboloids of Beauravia diffusa for treatment of kidney disorders. We identified about 45 metaboloids in them, and then there are different we have seen the variability among them, and after that we did the ADME toxicological profiling and for their effect on kidney disorders. And after that, what we did assess the targets, which are the pathophysiological targets responsible for kidney disorders, and then find out the major components, major 10 out of them, that is aposinin, oleic acid, linalool, and valerates, phenyl propionic acid, limonene, and phenyldeconoic acid, palmitate, linoleic acid, and menthol were having most active targets, and that's how we did different metabolomic work in different diseases and do in our laboratory. And thank you very much for hearing me. That's all from my side, and I think I must be in time.
Oh, thank you, Dr. Are you done? Okay. Thank you, Prof. Said. Okay. Can you unshare the screen?
Yeah.
Okay. Right. Okay. Yeah, we have a bit of a problem with the lagging, so the audience are still in the looking at your presentation, your slides. I looked at the Slido link and there's no questions at the moment. Just give them a few minutes to see if anybody wants to post a question. But yeah, I'm very impressed with the amount of work that your group has done in this area of metabolomics. It's a very challenging field, but it's actually a way forward for herbal medicine to be more relevant to human health. Because this is an opportunity for us to bring traditional medicine into healthcare in a better way and in a more effective manner. So, are we done? Oh yeah, they're on the last slide. So I don't know whether they actually lagging on our side as well. They're not seeing us. Okay. So actually, we are very, very much delayed in time. So what I'm going to do is I'm sure there are many questions. So I'm going to ask the participants to actually post their questions in the Slido chat box. Then we will forward the questions to you. So in recognition or in appreciation of your presentation today, I would like to, the organizing committee is very happy to present you with a certificate. The certificate. Okay, so there we are.
Thank you very much.
Okay, thank you very much, Prof. Syed Ahmad. I look forward to see you physically, in face-to-face, so that we can discuss more about this area of work.
Okay, thank you very much.
All right. So I think with that, Mumtaz, you are ready to introduce Prof. Malhalan with her presentation, perhaps?
Yeah, Prof.
Okay,
Thank you, Prof.
Thank you.
Okay. So I would like to introduce our next speakers is Professor Dalsy Malhalan, okay, from University of Surrey in United Kingdoms. Currently, she is head of the department chemistry, head of chemistry in department chemistry. Her particular interests lie in the phytochemistry of Hanisiaceae, Amaryllisaceae, and Euphorbiaceae. She has published over 250 papers in the field of phytochemistry, synthetic and analytical chemistry. Today, Prof. Dalsy will give a talk on homoisoflavonoids for the treatments of retinopathy. Without further delay, I would like to invite Professor Delphi. Prof.
Thank you. I am here. Are you going to use my recorded video or shall I do it live?
Okay, you want me to share the recorded video? I'll try first, okay?
Okay, well, let me first just thank the organizers for organizing us today, inviting me to the conference and this organization, which is very difficult to do under our present circumstances. And it's been a delightful morning in the United Kingdom, participating this morning. Thank you. Yes, if you share my pre-recorded video, then I know I'm right to time.
Yeah,
Thank
sure,
you.
Prof. I will try to share your video, yeah? Yeah. Okay, can you see?
Yes, it's a little bit small. Can you make it bigger at all?
Oh, yeah, sure.
Wonderful. Oh dear, the sound's not very good. Shall I do it live? Is the sound working?
I'm sorry, Prof. Did you ask? I'm sorry. You want to do the live?
I was wondering, I can't hear it from where I am, but if you can hear it, that's fine. Go ahead.
Okay.
I can't hear it.
Is it okay? Can you hear me, Prof?
Yes, I can hear you, but I couldn't hear the presentation. I was wondering if you could hear it there. If you can, that's fine.
Oh, okay, because the audience said the sound is not really clear. So is it possible for you to do the live or how is it?
Of course. Yes, of course.
Yeah. Okay, then I stop sharing, yeah?
I'll try and catch up on some time.
Sure, Prof.
Okay, good.
Because the audience said the sound is not really clear.
Excellent. So it's just going to take me a little moment to get here. Good morning. Good afternoon to you. It's good morning in England where I am. Today I'm going to talk about the potential of naturally occurring and synthetic homoisoflavonoids for the treatment of retinopathy. I hope you can hear me.
Yes,
Very
Prof.
well. Very well. Thank you.
Very good.
So since the early 1990s, I've been working on a plant family called the Hyacinthacea. Actually, that's not quite true because they're no longer called the Hyacinthacea. Botanists seem to like to change plant names, so they've now put these plants into the Asparagaceae. But for our purposes, we still call them the Hyacinthacea. And the reason why we look at these plants should be quite obvious from our review and the front page of Natural Product Reports. You see how beautiful these plants are, and this originally attracted us to these species. And we've found a lot of uses for extracts from these plants in the treatment of retinopathy of prematurity and age-related macular degeneration, as COX-2 inhibitors, for their anti-cancer activities, for their vasodilatory activities, all related to the treating of aging conditions. The Hyacinthacea is divided up into various sub-families and all have particular chemical profiles. So the Hyacinthoides produce mainly homoisoflavonoids. Well, you're all familiar, I'm sure, with flavonoids, but not many people know what a homoisoflavonoid is. A homoisoflavonoid is a flavonoid that's got an extra carbon there at C9. Usually there's a hydrogen here, and if there's a hydrogen here, it's got the R configuration in the Hyacinthoides. And you can get a range of different types with the double bonds here, and even with the C9 being incorporated into a four-membered ring. And that's important for later on when we talk about some of the activities. The Erginioides subfamily contain these bufadienolides, cardiac toxins, and we've isolated a lot of those. Very rarely homoisoflavonoids, but quite surprisingly, when you have a homoisoflavonoid with a hydrogen here, where it's R in this group, generally they are S there. The ornithogoides have got cardenolides, and again, if they've got homoisoflavonoids, they've got the S configuration here at C3, and that's very useful for our screening program to get both of the different enantiomers. One of our early plants we looked at was Ledeborio vatifolia. This is a South African traditional medicine. It's used to treat sprains, bruises, general external or muscular pain. You rub the bulb onto it and it helps with inflammation. And as is normal with these species, we got a whole mixture of compounds. Some of the homoisoflavonoids with the four-membered ring, some with double bonds here, some with H's or OH's there, and their precursors, chalcones, dihydrochalcones, and even some xanthones. You see quite a complex mixture. So based on their traditional activity, what we did was we screened them for COX-1 and COX-2 activity. Now, COX-1 is important in the normal functioning of the body, so you don't want your compound to inhibit COX-1, but you want it to inhibit COX-2, which is, put it simply, responsible for the inflammation. And if you look at some of these compounds and the active ones I put the structures on, we got total inhibition, zero activity of some of these compounds. And they were still allowed quite a lot of COX-1 activity, up to 73% for compound 10, this particular homoisoflavonoid down here. Now, we usually have quite a lot of compounds available for screening, and because we were setting up the screen, we took some norlignans that had come from related Hyacinthaceae, Dromiopsis species, norlignans. So it's a norlignan because there's one phenylpropanoid unit, here's the other, and it's lost its third carbon there. So hence the word norlignan. So we put these into the screen, and what we found that compound 1 was extremely active. It totally inhibited COX-2. but very low inhibition of COX-1. So we found this quite exciting, and we decided to try and synthesize a library of these compounds. So we decided to use some basic first-year chemistry, try an aldol reaction to get this compound here, add an R group using the vinyl Grignard reagent, reduction of this carbonyl to give an alcohol, which could then be reduced to give us the molecule we wanted. And of course, it didn't work. Why? We found out that, of course, if you've got this highly activating OH group on your starting material, the aldol reaction doesn't work. But if you change that starting material, for example, to a methoxy, you can get a really good reaction to occur in high yields. So we did this reaction, dimethoxy. It doesn't matter if you've got an OH on this side of the on this participant in the reaction, as long as you've got methoxy groups on your aldehyde. And this gives you this chalcone and 64% yield. The Grignard reaction was surprisingly easy to do. Often we have problems with Grignards. 55% yield. Reduction of the carbonyl worked very well, 61% yield. And then we had to dehydrate it to get our double bond here. If we use the standard DMSO, heating it up, we got an 81% yield. But if we use sulfuric acid, we had a very interesting reaction, 53% yield, but we got a cyclized product. We formed this cyclized phenyl indane. Phenyl indanes, there's very little in the literature on making phenyl indanes because they're really difficult to make. So this is a project we're continuing with, trying to find more of these compounds to put into our anti-inflammatory screen. Right. The next part of my, seem to be missing a slide somehow. We've been working on the treatment of retinopathy. Retinopathy relates to problems with the retina in the eye. The retina is the light-sensitive layer that allows you to see. And for you to have good vision, that retina must be smooth. Okay. So any damage to the eye caused by unwanted blood vessel growth leads to sight impairment and blindness. So if you have blood vessels growing abnormally, they end up giving you a bumpy retina. So your vision becomes distorted and can lead to blindness. There's several types of retinopathy. One of the more common is retinopathy of prematurity, premature babies, the most common cause of blindness in children. One of the most famous people suffering from this is Stevie Wonder, the musician. And this was only found out in about the 1940s when the incubator was developed for premature babies. And this was a high oxygen environment, put an extra high concentration of oxygen in to try and help the premature babies. But they didn't know that the blood vessels in the retina develop quite late in the development of the fetus, and the high oxygen environment caused abnormalities in the blood vessel formation. The second type is age-related macular degeneration, a growing problem with the aging population. Millions in the world. This is the amount in the UK suffering from this problem. And diabetes, a growing concern, again with the aging population and obesity problems. So let's have a look at what the eye looks like when you have one of these diseases. So this is a healthy eye, regular blood vessels, no blood vessel leakage. In age-related macular degeneration, you've got leaky blood vessels. Okay, of course, that's going to impair your vision. Diabetics have abnormal blood vessel growth and also some leakage. And in prematurity, abnormal blood vessels, far too many blood vessels growing to impair vision. So what are the current treatments? Photocoagulation, photodynamic therapy, and most importantly, anti-VEGF biologics. These have got side effects. They're very expensive, and a large proportion of the patients don't respond well. But the worst thing about these treatments is they all need to be injected into the eye. Of course, this is unpleasant for the patient. You have to go to a hospital to have this done, so it's time consuming. So there's a critical need for novel therapies, and in particular, eye drop therapies. So in 2004, these researchers in Planta Medica, get that out the way, in Planta Medica reported this particular molecule, cremastrenone. They didn't determine the configuration at C3, but they isolated as an active component and it prevented angiogenesis. Collaborators in Indianapolis in the USA looked in the literature to find out who else had isolated this compound. And there were two previous reports, Adinolfi and his group in '87 and my group in 1999. So Professor Corson asked me if I had any homoisoflavonoids to put into his screen. And just to show you what cremastrenone does in premature mice. Okay, so this is the scenario with cremastrenone. This is without cremastrenone. And you look at the difference in tubule formations. Okay, these tubules are formed by HREC cells that we'll be talking about, which are implicated in this abnormal blood vessel growth. So if you treat the premature mouse with cremastrenone, you have far fewer of these tubes forming than in the untreated case. And if you look at the retina, very few abnormal blood vessels compared to the untreated case. So this looked all very promising. And so if you can stop the cell proliferation of these HREC cells, human retinal endothelial cells, you can stop the blood vessel formation, you can treat retinopathy. Now you need to have specificity. The compounds need to be specific against these HREC cells, but shouldn't affect normal eye tissue. And so we always test against ARPE19s, normal human retinal pigmental cells, just to show that the compounds won't affect normal eye tissue. We also did cell migration assays and tube formation assays in the USA. So this is our first screen really from compounds from my group and Professor Sarah Seon-young from South Korea. We sent in a lot of our compounds to Indianapolis. And here's one of our compounds. We got a really good GR50, 0.217 micromolar. Here's another one of ours. Here are some of the synthetic ones at the bottom from the South Korean laboratory. So these all looked very, very promising. Now, there is a problem. We get a range of these homoisoflavonoids from natural sources. But as you'll remember from a previous slide, they occur as very complex mixtures, take a lot of time to separate, and many of these plant species that produce them are endangered. So there's very little likelihood of ever getting enough from the wild for commercialization. However, these compounds are small, simple to make, and we're busy making a massive library to screen them. So here's one of our syntheses we use, standard Friedel-Crafts type reaction. So you take an acid chloride, you treat it with your aromatic compound, you get the bond formation occurring under these conditions to give you the dihydrochalcone. Now you may be wondering, where does this extra carbon come from in homoisoflavonoids compared to normal flavonoids? And it's quite simply that it comes from a methyl group here. A methoxy group at that position, that carbon becomes incorporated into the ring in the biosynthesis. So if you use a reagent that will methylate that position and then do it under conditions for cyclization, you can produce homoisoflavonoids with the double bond in that position in about 60% yield. And we have synthesized a whole library with different hydroxy, methoxy groups in different positions. A second way of doing this is as follows. Firstly, you make an ether here using these reagents. You make this ether. You then treat it with polyphosphoric acid at 80 degrees to get cyclization occurring. And then treat this with an aldehyde, piperidine 80 degrees C, and you can produce a range of compounds again for the library. And some of the ones we have made have got methoxys, sulfur-containing compounds, etc. You can make a whole range of them. So let's take one of them. This one we produced with the double bond here, compound 44, methoxy and hydroxy here. And then we did a whole lot of reactions to see what happens when we treat that double bond. So firstly, we did a very standard reduction, palladium, carbon, hydrogen, atmospheric pressure, room temperature. And we've got a very surprising result. We lost the carbonyl. Not only did we lose the carbonyl, but we hydrogenated the double bond. We got a mixture that's at C3 there. If we use the Wilkinson's catalyst, what happens was we just reduced the double bond. If we used a chiral reagent, this iridium catalyst, we got specifically, that's the R form. And if we use trimethyl chloride, all that happened was we got rid of a methoxy group, converted to the hydroxy group, but there. So we put this into a test against these HREC cells, and here's our most active compound, compound 46. Compound 46, it's the one that it had the carbonyl and the double bond reduced on the previous slide. So that was quite an enormous improvement on the natural products. 45, one of the other synthesized ones, this one here, all those methoxy groups, was also quite active. And here I've got a range of ones that came from natural sources. One of the most active was this one, RCB1, which you'll notice has got the different configuration at C3. And this comes from a Madagascan species. So our next step was to take some of these compounds and treat them for their selectivity to see what they do against normal tissue. So we repeated the HREC experiment, and we did the ARP19 as well, repeated. Here's our most active one. Unfortunately, you'll see here that it wasn't specific at all. It killed off normal R cells as well. So it's not a good lead, but it's given us an idea that removing that carbonyl and producing a library of related compounds could be quite good. But our second most active compound, 44, this one, was really very selective, giving us some clues as to where we should go next. And we did the usual tests. These are our compound numbers. The black is the test against the problematic HREC cells. The blue is the ARPE19s we usually use. So you want a big gap between killing off HREC cells and affecting normal cell tissue. So this was quite a good candidate. This one not so good because it kills off both tissues. And so we've done a lot of this sort of experiments in Indianapolis. You can also look at the tube formation of these HREC cells. So this is normal, the control in DMSO. And here we've taken some of our natural products, 13, 27, and 28, some of our more active compounds. You'll see that at 10 micromolar, very, very little tube formation is occurring. These ones are also really active, and you can quantify this activity. And if we look at our most active compound, we're now down in the nanomolar range. You're seeing very little tube formation occurring compared to the standard in DMSO. Right, now you might ask, why are we making such a big library of compounds? Well, because very small changes can make an enormous effect on the activity and the selectivity. So let's look at compounds 1 and compounds 2. These are from natural sources. 3S configuration. Both got 3S configurations. How they differ, this one's got a methoxy there and a hydroxy there, and these are just switched around. So you might expect similar activity. Well, if you look at the activity against the HREC problem cells, you'll notice that compound 1 is about 4 times more active than compound 2. So what about the specificity? You'll see that compound 1 is not very specific at all. It affects other eye cell lines, whereas compound 2 is far more specific than compound 1. Just that minor change. What about comparing the 3S with the 3R? Well, the 3S have an activity GR50 of 0.13, and these are repeated experiments. The 3R, 93.2. So the 3S form from Madagascar rhodocodone is 700 times more active than the 3R form. We were just fortunate to get both out of different extracts for comparison. So you see, these are really nice lead compounds. We've just got a grant from the UK Macular Society to carry on this work for 3 years, synthesizing a big library with the aim of getting about 3 compounds onto proper clinical trials. To show you some other species from this family, Erginavia is from the Erginoideae, containing mainly bufadienolides, and these compounds have got really good anti-cancer potential. I'll just show you one of them. We send all our compounds to the NCI in the USA, and here we're showing activity against quite a number of their cell lines. These are all the cell lines they use in their panel, and quite good selectivity as well. This is the particular compound being screened, nice bufadienolide with a acetylated sugar on it. So good potential there, and we're carrying on with that work. And finally, vasodilating properties of these compounds. So this is done by my colleague, Professor Fabio Fusi in Siena. You take rat aorta muscle, you clamp it here, and these are attached by wires, which can detect any contraction or relaxation. And these were the compounds that we selected. You'll notice 4 homoisoflavonoids, one was more standard one, 3 that had 4-membered rings, and some other compounds that we had from the same extract. So what you do here is you pre-contract the rat aorta muscle with K70, sorry, this keeps getting in the way, sorry, K60, you contract them, and then you measure, you add your sample and you measure the relaxation of the muscle. And so you can see these 3 compounds here cause the muscle to relax quite remarkably compared to the other compounds. And if you look at what these compounds are, they happen to be the 3 that had this homoisoflavonoid with a 4-membered ring with the extra carbon incorporated, C9 incorporated into that ring structure. So this was quite exciting news that it was only the ones with a 4-membered ring that did this. And we tried this in several scenarios, taking off the endothelium or leaving the endothelium on, and we got the same sorts of results. So these compounds, the ones with the 4-membered ring, have significant vasodilating effects. They've got antispasmodic and spasmolytic activity found to be in other experiments via a negative modulation of the plasmalemmal calcium influx responsible for contraction of the muscle. Tried various other experiments, and we showed that the blockage of the channels occurs by the voltage operated calcium channels, and they work at the same level as the drug nifedipine. And so these are, again, showing great potential for treatment of these types of conditions. So I'd like to thank you for your attention. I would like to thank the organizers and the coordinators of the session for all their help, and all of you for listening. And I'd like to thank my students and postdocs at Surrey, Professor Crouch, my longtime botanist collaborator, my collaborators at the University of Graz who grow up bulbs for me and provide them, Professor Fabio for doing the last part of the screening for me, and the group at Indianapolis for hosting my students and undertaking the screening, the NCI for screening all our compounds for anti-cancer activity. I'd like to thank our funders. And just to show you where we are at Surrey, there's London. We're 45 minutes southwest of London by train. There's Heathrow Airport. There's the other big airport, Gatwick. So we're halfway in between the two. So always looking to welcome visitors to our department. And thank you for listening to my talk, and I'm happy to answer any questions.
Thank you very much, Prof. Delphi. It's a very nice talk. I mean, that's very nice place as well. We do like the weather in UK. Anyway, so I will ask any questions. I will see on the chat box. Is there any question for you? Okay, so thank you very much for very interesting presentation for Delphi from Dr. Ati. And then we do not have any question on the chat box. So is it the presenter? Do you have any question to Prof. Delphi? I open the floor. No questions? Dr. Ati, do you have any questions? Prof. Kozira? No? Okay, that means, yeah, very clear.
Beautiful presentations, Prof. Mahalan, Delphi. I just have one teeny-weeny question. Perhaps you can, enlighten me about the reception of funding agencies in the European countries with regards to research that deals with isolation of compounds from plants, because in our country, in our side of the world, such projects now receives a little, not little, but less preference or less priority due to several aspects, because this is a very, very intensive research which requires a lot of funding, etc. So what is the reception of the European funding agencies in Europe?
It's difficult. When I worked in South Africa, I had far greater funding than I've had since moving to England. I was fortunate when I first moved here 15 years ago to get a big EU grant, international EU grant, where we've developed a good agrochemical. But for the medicinal plant area, it's quite difficult. I've been really fortunate to get this Macular Society grant recently after many attempts at different funding agencies. Previous to that, we were doing the work just on the money that comes with PhD students and things like that. The one that's been quite useful recently and has led to lots number of publication is the UK's Royal Society travel grants, which enables young students from all over the world, and it's been mainly from Africa we've been applying for. I think we've had 6 grants enabling young researchers to come over to my lab, and I've been able to go and do workshops in their home countries, and that's worked very well. But major grants in medicinal plant research, it's really difficult here as well. It's such a pity because there's such potential.
Yeah, now with the pandemic, there's not only vaccine that's needed by the world, they also need new antiviral drugs and other drugs, like what Prof. Kim has actually presented on orphan disease, So who's going to take care of needs, especially for the third world countries? Yeah, so with that, thank you for answering my question. So maybe over to Mumtaz to finalize.
Okay, Prof. That means everywhere we face the same problem. Yeah. Okay. So since we are running out of time, so I think maybe the host want to flash the e-certificates. Okay, this is as a token of appreciation for you, Prof. Delphi. Thank you very much for the nice talk. Okay, without we end, before we end our sessions, is it possible for all the presenters to switch on the camera? Then we can take the pictures. And maybe can stop sharing the,
Let me make myself presentable. Okay.
So, is it possible to switch on the camera? Professor Kim?
Sorry, I have a problem with my camera.
Okay, no problem, because you have very nice profile picture.
Thank you.
Prof.
Anyway.
Kim, are you there?
Prof. Kim is on. Can you share your video? Give it some time.
And you switch on the camera.
I think he's there.
Can hear some, okay, he's muted. Okay, unmute. But
Video.
we can't hear you, Prof.
He needs the video. Uh-huh.
Yeah.
Hi again.
Okay, so we can take a picture. So everybody ready? 1, 2, 3. Okay, one more. Freestyle. One more. 1, 2, 3. Okay, thank you very much, everyone. So enjoy your evening. Have a nice evening. So we'll see you tomorrow. Bye.
Yes. Thank you.
Bye.
Bye.
Thank you very
Bye.
much.
Selamat
Professor
sore.
Fiza and Dr.
Selamat sore,
Thank
ya.
you very much. See you
Thank you.
tomorrow.
Bye-bye.
Yeah. Okay, bye-bye. Thank you, Professor Dulcie.
Bye-bye. Nice to see you.
Nice to see
Okay,
you.
no problem.
Sorry, I cannot open my camera.
Tapi